I'm so grateful to be here. I'm a PhD scientist, so I don't get to talk with or interact with patients very often. And it's really meaningful for me because I'm like working in my lab and it's, easy to get caught up in what we're doing in the lab and sort of lose sight of why we're doing this. So it's really, really meaningful for me to be here with you guys and to talk to you a little bit about like what we currently know about MPN biology, which is actually quite a bit, but there's still a lot we don't know. So those are just my disclosures. So um, we'll start really from the beginning. So I think everybody probably in this room understands that MPN is a disorder of normal hematopoiesis, which is the process by which our body makes blood cells. And so what actually got me interested in studying blood was that I find it to be such a cool system that our entire blood system comes from one single cell. And that's this hematopoietic stem cell at the top here. So this is the cell that like lives in our bone marrow and it receives signals to make all the other cells we need that comprise our immune system, and kind of every other cell that makes up our blood comes from that one single cell. And so in MPNs and in other myeloid malignancies, we sort of ignore, there are two branches, lymphoid and myeloid, right? We sort of ignore the lymphoid branch. That's starting to change, as I'll mention at the end, but really what we're focused on is the myeloid branch and why in MPNs we have myeloproliferation, which is overproduction of myeloid cells. And so, again, I think probably everybody in this room is aware, but I'll just sort of go through what MPNs are. So they arise, and this is important, they arise in that hematopoietic stem cell that makes all the rest of your blood. And what happens is you acquire a mutation in that hematopoietic stem cell, and that hematopoietic stem cell has daughter cells and all sorts of progeny that carry that mutation. And that's what ultimately causes these diseases. And so we know that MPNs, classical MPNs, are comprised of three distinct diseases. And they're all characterized by the overproduction of a different type of blood cell. So if you have too many red blood cells, you have a disease called polycythemia vera, or PV. If you have too many platelets, or the cells that make platelets, which are called megakaryocytes, this will give rise to a disease called essential thrombocythemia, or ET. And then finally, the most sort of complex MPN is in part initially characterized by if you have too many white blood cells. But really what happens when you have this disease, primary myelofibrosis, is you start to get scarring of your bone marrow. And we actually are still trying to understand the biology of that, like what causes that scarring, what are the cell types involved in that scarring. And then, of course, as we all know, and probably is a source of anxiety for a lot of people, there is a small chance that any of these can transform to acute myeloid leukemia. And this is a big area of research in the MPN field. We really are trying to understand, why do some people transform? Why do most people not transform? What causes those events? And so I love the history of kind of blood cancer in general, but especially of MPNs. And so I always put this guy up who's really like a hero to us, because he was the first person to understand that these diseases had all kind of some common cause. And what he noticed was that there were these people who were having kind of evidence of myeloproliferation or overproduction of myeloid cells. And he hypothesized that it was due to one common thing. He didn't know what it was at the time, but he called it an undiscovered stimulus. So what's that undiscovered stimulus? This was in 1951, and it took us 54 years, but we finally figured out that one of those undiscovered stimuli was mutations in JAK2. And so this was like a huge boon to the field, because we finally understood these mysterious diseases that are causing overproduction of myeloid cells are caused by a mutation in a gene. And so this was a big deal, and you can see these are 4 different groups published this at the same time. And it really drove the field forward in a lot of ways. And so now we know 75 years later, we pretty much know the mutational landscape of these diseases. So we now know that there are 3 driver mutations, driver meaning you only need that mutation to get the disease. So it drives the disease. So we have mutations in JAK2, which are the most common MPN mutation. My favorite mutation, CALR, this is the one I've dedicated the last decade plus of my life to studying, is the second most common mutation. And then a small fraction of patients have mutations in the thrombopoietin receptor MPL. And then we still have this gray, piece of the pie that we call triple negative. That's, people differ on kind of what they think that might be. But for the most part, we really do have a good understanding of the mutations that drive these diseases. And like Ruben said, if we understand the biology, we can develop targets for therapy. And knowing these mutations has gotten us quite a ways in terms of developing therapy, which I'll talk about in a little bit. So JAK2 mutations and MPL mutations are what we call activating mutations. And this just means that typically these are regulated proteins that receive signals to turn on and receive signals to turn off. And the mutations in JAK2 and MPL are activating in that they're always on. They have no off switch. And what the result of that is, which I'll talk about in a few slides, is that they keep telling the cells, grow, proliferate. They're never off. And so it's really kind of remarkable, actually, that this all happens because of one base pair change in the DNA. So here you see JAK2 wild type on the top. These triplets are DNA codons, and at the bottom, this is the amino acid that encodes that triplet DNA. And so the most common JAK2 mutation is JAK2 V617F. And so how that happens is you have this sequence GTC in the normal JAK2, and that G somehow gets changed to a T. And that one base pair change causes a valine amino acid, a V, to become a phenylalanine or an F. And that's at codon 617, so JAK2 V617F. And MPL is similar. So MPL is a receptor that lives on the cell surface. So its regulation is a little bit more complicated than JAK2, but it's a similar mechanism where you have, if you look here, TGG, which is a W, and that middle G gets switched to a T and it becomes an L. So this is at codon 515. This is MPL W515L. There's also MPL W515K, but similar mechanism where it's just one base pair change. And it causes that protein to always be on. So CALR, I'm a middle child, and I think of CALR as like the middle child of the mutations because it's very different. It's very unique. So CALR is not like this at all. So CALR mutations involve big insertions or deletions of lots of base pairs usually. And so I'm going to give you 2 examples because there are 2 types of CALR mutations, type 1 and type 2, that are the most common CALR mutations. So if you look at this CALR wild type sequence here, what I want you to notice is I've marked in blue all of these E's and D's, which are negatively charged amino acids. And the reason those are there in the CALR sequence is because they bind calcium. And that's a really important role for this protein, just to keep cells functioning normally. And so what happens when you have CALR mutations is, so this is the type 1 mutation, which is a deletion of 52 base pairs. So you lose those 52 base pairs, and those that were after come join up where those 52 base pairs were. And what happens as a result of this is the amino acids completely change, and now they're positively charged. So now you have a lot of R's and K's, which are positively charged amino acids. Positively charged amino acids can't bind calcium, so you lose that function of the protein, and that has all sorts of effects on the cell. And then the type 2 CALR mutation is not a big deletion, but instead an insertion of base pairs. And so what happens in the type 2 mutation is you get this addition of these 5 base pairs that once again change the amino acids. So you get, again, a lot of R's and K's at the end, which are positively charged. But I do want you to see, like, the type 2 mutation still has a lot of those blue negatively charged amino acids. So although there are commonalities between type 1 CALR and type 2 CALR, what we've really started to recognize and appreciate in the last several years is they're actually pretty different in terms of what they need, how they drive an MPN. And that's like what my lab studies. And so just to drive the point home of how unique CALR mutations are, they create a completely new protein sequence at the end of the protein. And this is going to become important when we talk about targeting CALR. And so if you just look at that red piece that's in what's called the C domain of the protein, that's a completely new sequence that you don't find anywhere else in your body. Like no other protein has that sequence. And so that makes it a great target for therapy because you can really go after just that mutant piece of the protein. And so this is what it looks like. So if you look at WT is wild type on the top, that's the sequence of the wild type protein. And then both the type 1 and type 2 have this. This is a 36 amino acid long, completely new C-terminal tail. So really unique mutation, unique protein. So what do all these 3 mutations have in common? They all activate the JAK-STAT pathway. And what's the JAK-STAT pathway? So the JAK-STAT pathway is what we call a cytokine signaling pathway. Cytokine is a growth factor that we all have in our body, and these pathways are very tightly regulated. So when a cell needs to grow, the correct cytokine will come in, bind to the receptor, activate it, and activate downstream signaling. And so how does this happen? So you have what are called, each of these purple things is a monomer, a receptor monomer. And in order to be activated, they need to join together and become what's called a dimer. And the way they join together is for a cytokine to bind. So the cytokine kind of acts like a glue to like bring them together and activate the receptor. And what that does then is you have these JAK proteins that are bound to the receptor inside the cell. And when the receptor monomers come together, the JAK proteins are now close enough to each other that they can activate each other and they can activate the receptor. And that's done by a process called phosphorylation, which is just a transferring of phosphates to certain residues on the proteins. And so when JAK activates itself and activates the receptor, what happens is you have these STAT proteins that are sort of floating around the cytosol, not the inside of the cell, not active. And once the receptor is phosphorylated with those yellow P's, those STATs can now bind to the receptor, and now they're close enough to JAK to also get activated by JAK. So JAK is activating itself, the receptor, and the STATs. And so just to drive home how important JAK is, because that will become important. So when the STATs are activated, they're then able to move through the cell into the nucleus, where they sit on DNA, and their whole job is to tell the cell, make us proliferate, make us grow, make us survive. And so you can imagine, like, this is an important process for like normal cells. When we need cells to grow, this happens, but it's also important that it's able to turn off, because when we have enough cells, it will be like, okay, we don't need there are, regulatory mechanisms that will say, okay, you can turn off. We don't need any more of you. And so what happens in an MPN is there's absolutely no off switch. And the reason there's no off switch is because all of this happens independent of cytokine binding. So remember, in a normal cell, you need that cytokine to activate the receptor. When you have MPN mutations, you don't need the cytokine. They're active regardless. And so this happens differently for each mutation. So in the case of JAK2 mutations, they are just active all the time. So they're always activating that receptor and activating what comes downstream of that receptor. CALR mutations are actually really interesting because they sort of act like a cytokine. They bind to the receptor and activate it, but they're not a cytokine. So they're able to bind to that receptor in a different spot where the cytokine would normally bind, but the receptor still views that as like, I'm being activated by a cytokine. And then MPL mutations themselves are just active all the time. So they don't need anything to bind to tell it to be active. And so in each case, you have what's called constitutive activation, always on. Constitutive is always on. And so when this is always on, you have this pathway of STAT binding to DNA, telling the cell to grow and proliferate all the time. You never have an off signal. And that's how these cells myeloproliferate or overproliferate, and we have too many of them. And so we have this concept in cancer biology called like Achilles heels of cancer cells, right? And I was interested in where this came from. So Achilles, I actually didn't know this until I was making these slides. So I didn't realize that the reason it's an Achilles heel is because Achilles' mom dipped him in the river as an infant and like didn't get one of his heels. And so that one heel was vulnerable.
Always blaming mom.
Exactly. And so what this means now to us is like it's a point of weakness, right? And so the JAK-STAT pathway was a very obvious thought for maybe this is an Achilles heel for MPNs. And the reason why it was an obvious thought for us is because A, it's active in all three mutations. So it must be really important because all three of them are finding a way to turn this pathway on. And it's driving cell proliferation and survival. So it stands to reason that if you stop that pathway, the cells might die, right? And so this was tested basically by making inhibitors of JAK2. And nope, wrong slide. Okay. And so what people did was to develop these small compounds that could bind to the JAK2 protein and turn it off, essentially switch it off. And the way that they tested this was to have cells that have MPN mutations, in this case JAK2, V6, and 7F, and you just treat the cells in a dish with this drug, and do they die? And do cells without the JAK2, V6, and 7F not die? So in this case, their control is BCR-ABL. And so what you'll see is that these two top lines, this is percent inhibition of cell growth. So it's a little bit counterintuitive. But the higher they go, the more they're dying, essentially. And so these two top cell lines have JAK2, V6, and 7F mutations. They're treated with the JAK inhibitor, and they're dying. Whereas the BCR-ABL cells, which don't have a JAK2, V6, and 7F mutation and don't have the same Achilles heel of JAK-STAT signaling, are not dying. And so basically the way this works is that if you don't have JAK activation, you don't have STAT activation, you don't have STAT sitting on the DNA telling the cell to grow, and so they die. And so the first JAK inhibitor, or the first targeted therapy that was approved for MPNs, was the JAK inhibitor ruxolitinib. And like I said, the way this works is it's a small little compound that binds to the JAK protein, it's this green thing here, and it changes its conformation to be in an off position. So it can't turn on. And so this was long before this was available for patients. There were years and years and years of testing this in the lab. And so in addition to the cell studies, it was also tested in mice. And this is typically how we do things. So we do things first in a dish, in cells. We do things in the mouse. We take patient cells and test them also in a dish to see, like, is this actually having activity against the cells we need it to have activity against? And so in this mouse study, what was shown, and sort of what I want to emphasize here, is you don't actually see, so on the right here is a survival curve. You don't actually see, like, I mean, it's significant, but the survival increase actually turns out not to be that remarkable. But what is remarkable on the left is how much this drug reduces spleen weight. And this remains true for people as well. So this drug is really, really, really good at getting rid of all those extra cells in your bloodstream that are making your spleen big, that are making you feel bad. And so this was actually like a really seminal discovery in the field. The problem with this drug is that it's not that effective at getting rid of that mutated stem cell, which causes the disease. And so that's what we're trying to figure out now, is how do we get rid of that mutated stem cell so that we can give people ruxolitinib, they can feel better, but we can also get rid of their mutated stem cell. So my favorite protein, CALR, is a completely different approach. So what we know about mutant CALR, as I said, is it binds to the thrombopoietin receptor and activates it. And so because it's on the surface of the cell, we can target this in a different way. We can use an antibody, and we can direct an antibody to just that mutated C-terminus of the protein. And this is really good because this means normal cells will not be affected at all. You have to have that mutated C-terminus in order for this drug to have any activity. And so the way that these are currently in trials, they're very new. So the way that this works is you have an antibody, which is this like claw thing that recognizes that mutant C-terminus and basically directs the cell to death. And it prevents the monomers from coming together, can't activate JAK-STAT signaling, the cell dies. On the right is another approach, which is called a bispecific antibody. That just means the antibody is specific for two things. Specific for the CALR mutant C-terminus, so it recognizes that. And it's also specific for a T cell. So it brings that CALR mutant cell, like by the antibody will bring it to a T cell, and the T cell will kill that cell. So a different mechanism, but similar approach. And so the way that this was discovered, again, back when I was a postdoc, like in 2013, my work and other people's work in the field was to figure out that mutant CALR is binding to MPL to activate JAK-STAT signaling. And that finding is what gave way to the development of these antibodies. So we like basic science is really where this all begins. And so what the people did who developed these mutant-specific antibodies were to take those cell models that we had used way back when to figure out how mutant CALR was working and treat those cells with this antibody. And again, you can see there's a lot of cell death in the treated cells, which are the blue squares. And there's zero cell death in cells that are not treated with the antibody, but in isotype control. So this is very specific, and it's really unique in that it's the first mutant-specific therapy And the reason we're able to have that is because of this unique mutation that has this really specific mutancy terminus. So what else is there? Are there other Achilles heels besides the JAK-STAT pathway? Because we know, as great as ruxolitinib have been, there are resistance mechanisms. There are ways sort of around the inhibition of JAK, and so we need other ways to target these diseases. And so the good news is that yes, there are lots of other Achilles heels. Some that have been subject to sort of early efforts to drug are, so we know JAK2, we know CALR, BCLXL, Naveen is here. BCLXL is maybe his CALR. MDM2. So these are all just other proteins that are somehow part of this network of signaling that's important for the cell that you can target and represent an Achilles heel, and you can kill the cell. And so there are there's a lot of hope. There's a lot of other options. And then beyond this, so what else are we working on? So what I'm going to list is just a small fraction of what MPN labs are working on right now to figure out new targets for therapy. But one area that my lab and many other labs are working on are, what are the other mutations that aren't JAK2, CALR, and MPL doing, like TET2, like ASXL1? How are these mutations contributing to the progression of the disease? And so we have many ways to do this. We have mouse models that have JAK2 and ASXL1 mutation, or CALR and TET2 mutations together. We have cells that we can put both mutations into. We have patient samples that have both mutations. And we can do so much biology on those models to understand, what is that other mutation doing? Is it necessary? What is biologically, how is it changing the cell? And then there are like other gene alterations that are not mutations, and these are called epigenetic alterations. So these change how the DNA behaves without changing its sequence. And these are actually very important in MPN pathogenesis and progression. So there's a big push to understand how these epigenetic factors contribute and can we target them. MPN is an very inflammatory disease. So how does this inflammation contribute? What are the pathways involved? What are the genes involved? What causes this inflammation, and how can we potentially target the inflammation? The bone marrow microenvironment. So your bone marrow, where this all is happening, is like a hodgepodge of so many different types of cells, not just blood cells. And so this is, again, an area that my lab is interested in. How do these cells interact with each other? How do the non-blood cells support the development of MPN in the blood cells, and vice versa? Like, what are they doing to each other? How does this bone marrow scarring in MF happen? Because the scarring is not in blood cells. And so that's a big area that my lab is very interested in. MPN cell metabolism, this has been a big area of investigation in solid tumors, and we're only just now starting to try to understand it in liquid tumors and blood cancers. How do MPN stem cells differentially utilize energy pathways in the cell to grow and survive? Can we target those to induce cell death? And then immune surveillance. So like I said, we kind of ignore the lymphoid branch, which is where a lot of the immune cells are. But we're now actually starting to understand that MPNs develop in part because immune cells aren't seeing those cells and getting rid of them. So why is that? What's causing that? What's causing them to lose that ability?